Welcome to Azad Bioacademy. Today I will talk about bacterial cell wall synthesis and associated antibiotics. Okay, so I will try to explain in molecular level. That means from the building block to the final cell wall synthesis. Okay, so let's go to the detail. First of all, I will try to give you some basics of bacterial cell wall. You know, the bacterial cell wall is wall, that means it is situated just outside of the cell, plasma cell membrane basically, and the cell wall, the basic component of the cell wall is peptide glycan. So peptide glycan, that means peptide and glycan, that means sugar and some amino acid assembling form is responsible for the formation of peptide glycan, and peptide glycan is the basic unit of bacterial cell wall. Okay, so how the peptides will be assembled within the cell and how it will exit from inside to outside, and how the sugar part will be associated and how it will contribute for the synthesis of bacterial cell wall. Okay, so come to the point. I will just try to explain in three categories. First of all, that is initial stage, or you can say initiation. That is basically cytoplasmic stage. Cytoplasmic stage. Okay, that means all the associated procedure or all those associated process will be within the cytoplasm. The second one is, you can say, elongation and transmigration, or you can say transport simply, and that is the membrane stage. Okay, and third one is extracellular. Third one is basically, it is peptidoglycan synthesis stage, or you can say cross-linking stage, and according to the location it is extracellular stage. Okay, so one after another I will try to explain. So firstly, I will go for initiation, that is cytoplasmic stage. What will happen inside the bacterial cell to produce the building block of the peptide glycan, basically all of the bacterial cell wall. Okay, so if you think this is, you can think like a wall which is basically built up of bricks and cement and sand. Okay, so such type of materials will be very necessary for the formation of bacterial cell wall. So come to the initial stage, that is first one, that is initiation, or you can say initial stage, or as it is happening within the cell cytoplasm. So this is the cytoplasmic stage. Okay, so come how and where it will produce. Okay, firstly, I will say glucose is the basic precursor molecule to produce the sugar part. Actually, just initially, just few minutes earlier, I said the sugar, that is glycan, and the peptide chain, that is amino acid assembling form. So, two types of sugar is the basic molecule to produce the basic skeleton of sugar chain. That is N-acetylmuramic acid and N-acetylglucosamine. Okay, this is the two basic sugar. and some amino acids that will form peptide, that is pentapeptide. I will go further. So it is glucose, or you can take also fructose, and it will follow some steps, and then it will turn into UDP, UDP N-acetyl glucosamine. Okay, that means UDP N-acetyl glucosamine comes from glucose. Okay, so here from several steps. So I will not go for that. So now, already I said N-acetyl glucosamine and N-acetyl muramic acid, two types of sugar, are very essential. So now, an enzyme will work on that molecule, and that will turn into NAM. So you can say that is mur A B, or another name is enol pyruvate transferase. Okay, that enzyme will work on it, and it will turn into UDP N-acetyl muramic acid. Okay, that means N-acetyl glucosamine will be converted into N-acetyl glucosamine, which will convert it into N-acetyl muramic acid by the help of the enzyme. Okay, so this enzyme is very much important. Some antibiotic will work on it. I will just explain in antibiotic stage. The next, that N-acetyl muramic acid will carry some amino acid, that is peptide. How the peptide will be added to the NAM sugar unit, then I will explain now. So now, one enzyme that is MUR C, that will add a peptide or amino acid actually to the NAM sugar. So now I will go to the structurally, I will try to explain. So now here is, you can think, this is a sugar unit, that is, you can say NAM, and one UDP is attached here, UDP. So this is UDP, okay, uridyl diphosphate. And one amino acid will attach to the N-acetyl muramic acid by the help of MUR-C enzyme. And then I will just draw like one amino acid. This amino acid name is L-alanine. L-alanine will attach to the UDP N-acetyl muramic acid. Okay. Now the another enzyme will work on it, that is the MUR-D, and that will add another amino acid to form a dipeptide, that is the N-acetyl muramic acid, and this is already attached with the UDP, and here one is the L-alanine, and now another amino acid will attach here by the help of MUR-D enzyme, that is D-glutamine. Okay, So now again another amino acid another enzyme will work, that is mu E, and it will add another amino acid to the dipeptide chain, which is attached with the N-acetyl muramic acid. Okay, so here the sugar part, and this is UDP is attached here, and one is your ALA, that means alanine. Then this is D-glutamine, and now third amino acid will attach here, that is L-lysine. Okay, L-lysine will attach with this dipeptide. That means now this UDP N-acetyl muramic acid is just carrying 3 amino acid, okay, as a just trimer of 3 amino acids: L-alanine, D-glutamine, and L-lysine. Okay, now another enzyme will work, that is the MUR-F, and this amino acid will attach 2 amino acid dimer at a time, and that amino acid dimer will form firstly. So I will show you, that is here the sugar unit, that is the N-acetyl muramic acid, and UDP is attached. Then here one is ALA, then D-glutamine, then L-lysine, okay, L-lysine, and by the help of MUR-F, it will add dipeptide, that is one is here, another one is, that is basically D-alanine and D-alanine. That is dimer form will attach here. And who will form dimer actually? In our human body, alanine is available there, but for bacterial cell, D-alanine is very much important. So L-alanine will turn into D-alanine, and then D-alanine will form a dimer, and then it will attach by the MUR-F. So here I will just try to show you in a short. That is, firstly you can take, that is L-alanine, okay. Then on enzyme will work here, that is racemase, and it will turn into D-alanine, okay. And another enzyme will work here, that is that is ligase, okay. Then what will happen? Another D-alanine will attach here with that D-alanine. So D-alanine attach will And this D-alanine also come from the same way. So D-alanine and D-alanine, it will produce D-ala and D-ala. Okay, so that will attach here by the help of this amino acid. Okay, so this is the one basic unit or molecule, basic molecule, which will contribute to form peptidic glycan. So this is the UDP N-acetyl muramic acid, which is just now carrying pentapeptide. So L-alanine, D-glutamine, L-lysine, then D-alanine, D-alanine dimer. It's just carrying pentapeptide. So some species of bacteria can carry 4 amino acid, 3 amino acid, but most of the cases pentapeptides are more common. Okay, so this is happening within the cytoplasm. Okay, so this cytoplasmic stage, we can say this is the initial stage, or you can say cytoplasmic stage, because this is happening in cytoplasm. Okay, so this is the cytoplasmic stage. So just I will try to just sum up by just keeping some basic things. That is, glucose will be converted fructose, then fructose on phosphate, then it will turn into UDP N-acetyl glucosamine. This is one of the basic unit. And another unit is the N-acetyl muramic acid. And N-acetyl glucosamine will turn into glucosamine by the help of enzyme mu-AB, or you can say the another name is enol pyruvate transferase. Okay, then it will produce UDP N-acetyl muramic acid. Then 5 different amino acids will attached by different enzymes. Okay, and everything is happening within the cytoplasm. So this is the initial stage, and this is the cytoplasmic stage.